Presented by Caltech.
So the first speaker is Holly Bick. She is from NYU, and the title of her talk is Evolution, Biogeography, and Systems Ecology in Microbial Eukaryotic Taxa. Holly.
All right. So to kick us off this morning, I'm going to talk about my research, which, as Sarkis mentioned, is focused on microbial eukaryote taxa. And I wanted to introduce my talk with a brief outline of my seminar. So I'll tell you a little bit about myself with a brief resume, followed by definition, phylogenies, and some genome projects, which are relevant to my talk. And then moving on to 3 stories about my research, which focuses on microbial eukaryotes in marine sediments. Firstly, the evolutionary history of nematodes. Secondly, using high-throughput sequencing to look at biogeography of these taxa. And finally, using systems ecology approaches and identifying biomarker taxa. So a bit about myself. I did my PhD finished my PhD in 2010, working at the Natural History Museum in London. And I focused on nematode taxonomy there in combination with molecular phylogenetics. So I come from a very traditional biological discipline. And over time, in my postdoctoral career, I've transitioned over to computational biology. So I really straddle the line between biology and computer science, and I work with non-computational biologists, benthic ecologists, as well as computer scientists and software engineers in my current research. So I did a postdoc at the University of New Hampshire, focusing on environmental sequencing, nematode genomics. Another postdoc at the UC Davis Genome Center, using metagenomics, software development, and bacterial microbiology, before doing a year-long fellowship in the UK at the University of Birmingham. And then currently I work at NYU on several projects related to urban metagenomics. So the term microbial eukaryotes is a pretty broad term. And I'll introduce a little bit about these taxa, but the take-home message is that they're extremely diverse and extremely abundant in pretty much every habitat on Earth. And my work focuses on free-living assemblages in soil, sediments, and even sea ice. And when I talk about size classes, typically this is defined by taxonomic protocols, which are traditional. And we're concentrating organisms onto a 45-micron sieve. So we get rid of the sediment and concentrate everything down. Typically, this doesn't retain anything bigger than about a millimeter in size. The protocols are designed to work on nematodes because of my background, but usually we capture the breadth of anything which fits in this size class. So fungi, protists, other minor metazoan phyla, and eggs and larval stages of larger organisms. And in terms of the number of species that we know about from morphological taxonomy, there is a huge taxonomic deficit. So there's estimated to be anywhere between 1 million to 100 million nematode species, but there's only about 4,000 described marine species. And most pertinent to this talk is that microbial eukaryotes are large enough to be physically manipulated. So we, under the microscope, we use an eyelash mounted on a glass rod to pick them out. But because it's labor intensive to do it this way, they're usually too small for most genome interests and efforts. So in addition to the taxonomic gap in our understanding of these organisms, there's a huge molecular gap where we just don't have any data in online databases. So to ask questions about evolution and biodiversity, my work focuses on microbial eukaryotes in marine sediments in particular. And this is an ecosystem itself, which is also understudied. So it's a huge environment. It's very difficult to sample. You have to go on a boat and put down heavy equipment to actually get sediment. And nematodes in this habitat are the dominant organisms. So their abundances range anywhere from 100,000 per meter squared all the way up to about 84 million per meter squared, depending on the productivity of the ecosystem. There is a huge abundance of species, so anywhere between 30 to 100 genera reported, 60 to 200 morphospecies reported per study, and these are sediment cores, which are only about 10 centimeters squared. So within a very small handful of sediment, you just have this huge diversity and huge abundance of these organisms. They're restricted in movement in sediments, so they can't get around as easily as organisms in pelagic habitats in the water column. And the assemblages are influenced by productivity of overlying surface water, so the organic flux, which is coming down into the sediments, as well as any sort of environmental disturbance which is happening to these ecosystems. So that includes pollution, such as oil spills, as well as bottom trawling efforts. So the sediments themselves are a very good case study for asking these big questions in evolution and biodiversity. Okay, to situate ourselves on the tree of life, so we are here in eukaryotes, and more specifically, we are this small slice of the eukaryotic tree of life known as nematodes. However, most genome sequencing happens in the part where I'm not going to be focusing my talk. So the databases currently are very biased towards bacteria and archaea, as well as the bioinformatics methods are also very biased. So a lot of times the methods that get developed aren't necessarily useful or relevant for the study of eukaryotic taxa. Okay. And to zoom in on the nematode tree of life a little bit, nematodes are a really interesting group. I know I'm biased because my background is nematode taxonomy, but they are a group that's great to work on because they have very deep evolutionary history and diverse life strategies. Unfortunately, the framework of the nematode tree of life is just largely based on a single gene. So we're still really behind the times when it comes to elucidating deep phylogenetic relationships in these clades. And C. elegans is in one of the crown clades. So there's 3 clades of nematodes. C. elegans is way up here. Which is where most genomic work happens. But the free-living marine species, which my work focuses on, are very divergent. They are early splitting lineages at the base of the nematode tree. And in terms of genome sequences, they're extremely distant from work on C. elegans and other nematode species. And most importantly, there's 0 genome data that exists for these marine species of nematodes. To illustrate that fact, so these are the genome sequences which are available online for nematodes. The majority are these bold black species, which are parasitic species, as well as model organisms such as Caenorhabditis, and free-living lineages which are relevant to the study of parasitic taxa. So this is what drives the genomics community for nematodes, is parasites and C. elegans. And unfortunately, for the study of natural ecosystems, it's frustrating because we just don't have genome data which is really relevant to the organisms which exist in marine sediments. So another, just to drive the point home a little further, the genome deficit in terms of eukaryotes is pretty dire. So nematodes, again, are a small slice of the eukaryotic genome work that's happening. Most projects that are funded focus on vertebrates, plants, and fungi. And there's only about 181 nematode genome projects in the genre which were on the previous slide. So there's a lot of questions that we can ask. And I will start my talk by telling some of the questions related to my first story, which is focused on the evolutionary history of nematodes. So because of the lack of molecular data, never mind genome data, we really don't have a good understanding of how this phylum has evolved over time. So we don't understand how the really diverse spectrum of morphological diversity in nematodes relates to underlying genetic diversity. So nematodes don't have many features. There's a lot of convergent evolution in morphology, and the data that has come out for molecular studies so far suggests that morphology is not a good indicator of underlying evolutionary relationships in nematodes. Secondly, in marine environments and terrestrial environments, we don't have a good sense of how common habitat transitions are, so between terrestrial and marine sites, and also shallow to deep water in the ocean. Nematodes are one of these taxa which are thought to very easily switch between habitats, so there's some molecular switch which allows them to live in terrestrial versus marine environments, and they can make that swap very easy. Another question which is relevant is, how can marine nematode genomes, if we were to add this data, how can it shed light on the evolution of terrestrial and parasitic species, which is where most of the research community is focused? Can we infer the evolutionary origin of nematodes? Did they originate in a terrestrial or marine environment? And finally, when we're looking at these vast understudied ecosystems like the deep sea, can we use phyla such as nematodes to reconstruct the historical evolutionary trajectory of this habitat as a source versus a sink habitat? Right, so I'm going to be talking about work which I finished during my PhD, and this is work that was published in 2010 in BMC Evolutionary Biology. So my PhD work, taking into account the molecular deficit for nematodes, focused on one of these early branching clades, marine nematodes, predatory species from the Enoplida. And during this work, I isolated 254 specimens from 37 genera, getting gene sequences representing full-length 18S, a fragment of the 28S 18S and also COX-1 genes. So this was really the first molecular data generated for some of these genera, and definitely for deep sea species that was really put into public databases. And one of the first questions we asked is whether or not the increased taxon sampling towards the base of the tree would enable us to get a better idea of which branch of nematodes split off first. But unfortunately, with a single gene with 18S, even adding a lot of taxa, it's not feasible at this stage with a single gene phylogeny to reconstruct early spridge. So the branching order was extremely unstable, despite doing lots of topology tests and using maximum likelihood Bayesian phylogenies. It's not enough, a single gene. Another question we asked is about the relationship between marine and terrestrial clades. And so in the phylogeny of this group of predatory nematodes, terrestrial species were recovered as sister groups to marine species, and this was consistent across the phylogenetic structure within this clade. Within marine environments, we were also asking about the relationship between deep sea and shallow water taxa. And again, it was we were seeing a sister relationship. So deep sea are the red clades here, and shallow water are the blue clades. And despite being in completely different habitats, beaches versus sediments that are 5,000 meters, the nematodes were consistently recovered as sister groups. So just zooming in on this phylogeny a little bit more, there really is no structure according to depth. So it, again, suggests frequent habitat transitions between deep sea and shallow water. And this was also published in BMC Evolutionary Biology in 2010 in a different paper. To shed some light on how morphology relates to genetics, an example of what we were finding is from the Phanodermatidae, and this was a group that has extremely little morphology, so this is what they look like from a taxonomic standpoint. They are very difficult to tell apart. There is a lot of morphological similarity between specimens, but the genetic information suggested a very strong diversity, of cryptic diversity in these specimens. And on top of that, despite having specimens from multiple geographic locations, there didn't seem to be any sort of structure according to ocean basins. So we had within a single clade, Antarctic, Indian Ocean, Pacific, Atlantic specimens, which were very close to each other in the phylogenetic topology. All right. On top of that, another thing that I found is the co-occurrence of multiple species within a genus. So within a single sediment core, again, just a handful of mud, it was possible to get up to 3 genetically divergent species within one site. And this is, again, focused on a single group of marine nematodes for a very small area of mud, really. So if we extrapolate this to the entire diversity of nematodes, it suggests that there is a lot of cryptic diversity, but also a lot of very closely related species in one environment. So this is, from an ecological and a biological standpoint, it raises a lot of questions about how so many species can coexist, and from an evolutionary standpoint, how they became distributed in that way. So during this work, what I found is that nuclear ribosomal RNA genes were really not sufficient for reconstructing deep splits in the nematode tree of life. However, habitat transitions appear to be common across clades, so between terrestrial and marine habitats, and also deep sea to shallow water. And there was really no phylogenetic clustering according to depth or ocean basin. On top of that, data that I didn't present also suggested low genetic divergence across vast distances. So sometimes it was possible to find nematodes within the same genus that had identical gene sequences for 28S, 18S, and COX-1 at sites which were 10,000 kilometers apart. So that's interesting from a standpoint of are they dispersing that far, or is this something interesting that's happening from a genomic evolution standpoint? Great. So this work is one of my main research areas. There's a lot of future work to do in this area, and the next steps which I'm going to be taking moving forward is really focusing on genome sequencing. So the time, because of developments in lab protocols, I think it's really, the time is really ripe to start getting full genome sequences, or at least gene-level contigs for some of these single specimens, and putting that into a phylogenomic approach to reconstruct deep phylogeny and expand taxon sampling for all marine clades. In addition to that, getting more samples. So if we want to get a global view of nematode diversity and distribution, leveraging samples from other habitats, chemosynthetic habitats, sea ice, and also depth ranges as well to build up a picture of global diversity. There's also nematode groups which I didn't focus on for my PhD work. So there's groups that are closer related to C. elegans in the clade 3, which are present in marine environments. These are typically bacterial feeders. So these are potentially more relevant to the study of model organisms and parasites because they are closer. And so characterizing evolutionary patterns in these groups is also a priority, as well as looking at groups which are endemic to the deep sea. So there are nematode lineages such as with quite interesting morphology. This is Manganonema, which has a really tiny head up here, which is only found in the deep sea, so it's never been found in shallow water. And putting these into the context of the nematode phylogeny and looking at the genetic diversity of these deep sea species is also a priority. And ultimately, the goal is to try to identify genomic signatures of marine lifestyles and extreme environments. So whether or not we can determine genomic signatures of productivity and free-living lifestyles in habitats such as the deep sea or sulfide-rich environments. Okay, so that's the end of the first part of my talk, and I'll move on to the second part, which focuses on biogeographic questions, and mainly the Bass-Becking's hypothesis, which is everything is everywhere, but the environment selects. And with tiny organisms that are abundant and diverse in habitats such as marine environments, one of the main questions is, is everything really everywhere? Are species cosmopolitan, as suggested by morphological literature, or are they really regionally restricted in terms of having a lot of cryptic genetic diversity? Another question is, how do community assemblage vary across space and time? So we haven't been very good as a community at looking at temporal variations. We don't really know about fluctuations in species, and maybe this is the key to understanding how so many species can coexist in one sediment core at the same time. There's also a lot of really compelling hypotheses from the classical literature, which are driven by morphological studies. So marine environments have putatively very high marine biodiversity. They've been reported to be peaks of species diversity at intermediate depths, as well as latitudinal gradients of diversity. And these are really interesting patterns and important patterns in marine environments, but they haven't been rigorously tested with molecular data. And another thing we want to know is whether or not these patterns are also consistent for all taxonomic groups. Typically, these hypotheses come from a very focused study of one organism, but it's relevant to our understanding of these ecosystems to look at patterns across nematodes, fungi, protists, and all different eukaryotic groups, as well as bacteria. And in terms of extreme environments, are there divergent evolutionary lineages and endemic hotspots in areas such as the deep sea, which have been cut off for a very long time and have very specific environmental constraints? So to move towards some of these global hypotheses, I really kind of got obsessed with these during my first postdoc at the University of New Hampshire, and that was the time that 454 sequencing was coming online, and I started moving away from single nematode studies and more towards environmental DNA workflows, because it provides a very rapid, quick way to test some of these major questions in biology. And so the workflow is pretty stabilized. This is consistent between bacterial studies as well. We start off with sample collection out in the environment. We collect frozen sediment and bring it back to the lab. The main difference for us is that we, for eukaryotic studies, we physically isolate organisms. So we're working with volumes of sediment that's about 400 to 500 milliliters, versus bacterial studies. Any sort of ribosomal RNA surveys for bacteria usually work with about 2 grams of sediment, which is put directly into the DNA extraction. So we don't do that. We physically concentrate eukaryotic organisms such as nematodes onto a sieve, and then we bring it through into DNA extraction from the bulk collection of worms.
How many different largest, small organisms would you find in that 500 mil?
Yeah, so anywhere between 100 to a few thousand usually. And then we typically pipette up from the whatever is in the microcentrifuge tube, so it's probably at least a couple hundred specimens, full specimens, but we also get a signature of any pieces of tissue, anything in gut tracts, so we get a wide spectrum of across eukaryotes.
So that's one per
It's 200 microliters that we pipette into the extractions itself, and whatever worms get sucked up in that. So after we sieve,
Are you able to put from the original sample?
oh, from the original sample. Yeah, I mean, it's a standard taxonomic protocol, so usually, yeah, any thousands potentially concentrated from 400 mils, depending on the productivity of the environment. Okay. After we extract environmental DNA, the next step is to amplify 18S ribosomal RNA because that's the most informative gene because of databases and phylogenetic utility, but we are also moving towards skipping this step and doing shotgun metagenomics in addition to 18S sequencing. Then we go on to sequence on Illumina machines nowadays, and the ultimate goal is to get this elegant picture of biogeographic patterns across space and time. And these data sets are huge, so there's a massive bottleneck in moving from sequencing over to this figure that you put in your manuscript. And this is a bottleneck both because of the volume of data that you have to process, but also because in the collaborations I work with benthic ecologists and taxonomists that don't have any computational training. So in terms of student time and getting collaborators to understand these data sets, there's also that training bottleneck, which makes things a lot harder. And to talk about some of the things we've been doing with these environmental studies, I'm going to focus on work we did published in 2012 in Molecular Ecology that was asking about how we can use environmental sequencing to look at global patterns in deep sea phylogeography. And for this study, we had 12 samples. And we had 5 samples in the deep sea Atlantic off the coast of Spain, 5 samples off the coast of California here, and we supplemented that with 2 additional sample sites in California and the Gulf of Mexico to represent vaguely geographically proximate sites. The great thing about environmental sequencing, which is not true of nematode morphological studies, is that we can essentially recover all taxa which are present in these samples, which we pipette up from the environmental DNA. So not only nematodes, but we recover about 20, upwards of 20 metazoan phyla, kinnorynchs, priapulids, tardigrades, anything. The primers are pretty good at recovering metazoa. But in addition to that, we also recover fungi, stromatopiles, algae, all across the eukaryotic tree of life, including deep protist lineages, although most likely with some biases in these more divergent groups. Unfortunately, the problem with working with eukaryote data is that the taxonomy is not always informative, so even our ribosomal RNA databases are pretty terrible for eukaryotes, and we just don't have as many things in there because there hasn't been this drive to generate molecular data for eukaryotes. But we also have these annotations which are frustrating when you're trying to link a sequence with a taxonomic ID. So we routinely get things which don't match to anything in the database, and by which I mean no good match, so nothing that has a 90% or more blast identity. And we get annotations which come back as environmental, which means that the sequence matches the database, but there's no informative taxonomy attached to that database sequence. So it's great because we can rapidly cover a breadth of eukaryotic taxa, but we can't always make sense of the data as much as we want to. And so for this study, one of the things we could do is we can use phylogenetic approaches, in this case unifrac distances and analysis in the CHIME pipeline to look at broader patterns without necessarily concentrating on the fine-scale taxonomy. And what we found in this study is that similar to what we expected from the morphological literature, we found a very strong divide between deep sea and shallow water sites. So even though Atlantic and Pacific samples were in completely separate ocean basins separated by 2 continents, the communities themselves at the molecular level were much more similar to each other than the communities from the geographically proximate sites. And again, this is supported by the morphological literature, so it was great to validate this with genomic information. Another interesting thing we found was 9 to 11% of OTUs, 18S OTUs, were reported to be putatively cosmopolitan, so present across deep sea sites or present from deep sea to shallow water. And it's interesting from a biogeographic perspective, but it's also potentially not the whole story, because one of the main issues with these data sets is that it's very difficult for us to correlate eukaryotic OTUs with biological species. So eukaryotes, unlike most bacteria I should say, have multiple ribosomal RNA gene copies, so from tens to 10,000s per genome, and there also exists intragenomic variation across these ribosomal RNA gene copies. So when you're doing surveys using ribosomal RNA data, you can contract and condense your OTUs, and you can use different cutoffs. But when you have a sequence out here that's kind of on the periphery, it's very difficult to tell whether that's a biologically true ribosomal RNA variant within a genome, or if it's a chimera or an error. And we did a little bit of work to validate this and explore these patterns further using nematode genome data that existed. So this was work I published in 2013. And if you look within a single genus of nematodes to give you a better idea of the copy variation, so within Caner abscetis itself, we found between 56 to 323 ribosomal RNA copies within one genus of nematodes. So there doesn't seem to be any pattern that governs how many copies these eukaryotes have. And it appears to be subject to selection. So for C. elegans mutation accumulation lines data, there was a twofold increase in ribosomal RNA copies after 400 generations. So clearly there's a lot more work to do in terms of what governs this ribosomal RNA variation and copy number. But in the meantime, it presents a huge problem for environmental studies where we don't have any controlled environment to test this. So the take-home message is that it's really difficult to infer biology when your data looks like this. This is output from CHIME, and when you give this to a taxonomist and you say, "Okay, give me some biology," it's a slow process and it's not always informative. So, then as we move forward and as the field moves forward, the next frontier is really trying to explore fine-scale patterns in these data sets, and the main way, which I think this is going to happen, is through data visualization tools. So I have made a concerted effort to try and come up with new ways to look at these data sets and also make it easier to give a taxonomist an OTU table or something from a computational pipeline and have them look at the data and kind of infer biology and ecology and get at more fine-scale patterns at what is happening in the environments that we're studying. So this was a grant I got funded as principal investigator through the Alfred P. Sloan Foundation. And this is a web-based framework. So if you go to finch.org, it's called the Finch Framework. In Google Chrome browser, and if you have a biome file from the CHIME pipeline, you can upload your data set and explore it. And visualization frameworks, the way I designed this project is really geared towards any sort of biological matrix. So although we moved forward on the initial stage of this project using OTU tables from the CHIME pipeline, we focused on the biome files as a JSON formatted file, which is computationally easy to handle. So essentially, if you are a researcher that has any type of data that can be represented as a matrix, so if you can have a tab delimited table or an Excel spreadsheet of data with accompanying metadata, it would be very easy to convert that into a JSON formatted file. The Biome Toolkit is online and pretty easy to use and install, and it comes prepackaged with the CHIME pipeline. And this is an open source project too, so it's the source code is available and reusable and out there. And so to give you a quick demo of this, again, we wanted to make it easy for people to upload data. So if you have a biome file that's formatted correctly, all the metadata is also embedded in that file. And essentially, you just go to the website, click a button to upload the file, and then the Finch reads the metadata and the sample data from the text file itself, and it parses the data and then brings you through into the visualization framework. So in a few clicks, you can start exploring your data sets. You can go down taxonomic hierarchies or gene ontologies by just clicking a button. We have search features implemented where you can look for your taxa of interest, maybe if this is a taxa with known ecological roles that you want to pinpoint in your data sets, and mainly also trying to interact with your data a little bit more, because existing bioinformatics pipelines make it very hard to just get more information from a visual image. So all of the features here, if you hover over anything, you'll get additional information about what that means. In this case, this is a bubble chart where OTU abundance is represented by circle size, so bigger circles are more abundant, and then clicking on a circle will let you look at the distribution of that OTU over sample sites. So this was built in conjunction with the Data Visualization Studio in Oakland, And currently we have 5 visualizations that are implemented, which can be used to explore data in different ways. So we used a mix of things that biologists like to see, like bar charts. Bar charts are always useful for publications, but also novel visualizations. That was a Sankey chart, which displays the taxonomic hierarchy or ontology. And the framework is mainly geared towards summarizing data in different ways. So very quickly being able to run something through a bioinformatics pipeline, upload it, visualize it, and maybe use that as a guide for your next round of sequencing, or using that as a jumping off point to go explore the literature a little bit further. So it works in Google Chrome right now, and it parses data on your local machine. So even though you're loading a file, you're not actually uploading anything to a web server. Everything is happening in your Google Chrome browser itself. And we took advantage of the ingrained JavaScript libraries, mainly IndexedDB. And I'm happy to talk about that a little bit further. But everything is on GitHub, so the source code's available and the documentation is up there as well. So I really think that visualization is one of the main ways forward where we can start taking advantage a little bit more of these huge environmental data sets. Okay, to summarize the second part of my talk, so I showed you a little bit about some of the early studies we did for environmental sequencing, and this was focused on deep sea and shallow water habitats. What we found is that at least with a limited sample set, the environmental data was supporting the reported biogeographic patterns of a strong divide between shallow water and deep sea habitats. But again, this was a very limited sample set, so as we move forward and work on other projects, we're generating a lot more data from a lot more geographic locations. Most sediment OTUs appear to be regionally restricted, but in these studies, there are usually a subset of cosmopolitan OTUs that are recovered, and the caveat being that it's cosmopolitan according to a 400 base pair fragment of the 18S ribosomal RNA gene. So that might not hold up with other data. Eukaryotic surveys have a lot of caveats. They're a lot harder to do than bacterial ribosomal RNA surveys because of the presence of intragenomic variation within the genome for ribosomal RNA loci, and also due to the database limitations. So eukaryotic databases are just generally terrible, and the annotations aren't as informative as they need to be. And finally, the visualization tools are probably the way forward in order to get around some of these limitations. They're not going to solve everything, but at least they'll let us explore fine-scale patterns a little bit more. And the rationale behind that is mainly just to use our ingrained powers of human cognition and pattern matching to look at these data sets in an abstract way and use that as a jumping-off point to pinpoint fine-scale patterns. So this is a really active area of my research, and I have other projects which are ongoing, we're in the thick of data analysis, specifically one focused on Arctic sediments. But as we move forward and go forward with these projects, we're trying to expand the set of data that we get. So ribosomal RNA surveys, because they're limited, metagenomics and targeted genome sequencing is the way forward. And I think it's the only way that we're going to be able to rigorously test some of the biogeographic hypotheses, and specifically look at how genetically divergent some of these specimens with identical 18S are. So it is possible to pick things out from sediments, and that's what we're doing. So we're sort of going back a little bit to the taxonomic protocols to try and link this environmental data with an organismal ID, because I think that's the way forward. I'm also in the process of writing a renewal grant for the Finch data visualization framework through the Sloan Foundation, and in the second stage of funding, the goal is really to develop better visualizations, specifically for phylogenetic visualizations, putting things onto a tree, developing public APIs so other researchers can link in with this framework, and also developing statistical analyses. So if you see something that looks interesting biologically, you can just push a button and be able to tell whether or not that is statistically significant. And finally, one of the main things as well, working on these data sets, is really thinking about how we can make bioinformatics pipelines more inclusive. So as we move towards expanded data sets, as we get metagenomic data, as we look at data from bacteria in conjunction with eukaryotes, one thing we can't really do right now is analyze that data all in one pipeline. So it's hard to do integrated omics analysis. But there's a lot of information in these data sets, and linking things like virus dynamics with eukaryotes and bacteria, I think, is a really interesting thing that could be pursued with better pipelines. All right. And with that, I'll move on to the last part of my talk, which is focused on systems ecology and the development of biomarker taxa. And so this is work, for this part of my talk, I'm mainly thinking about things in the context of the Deepwater Horizon spill in the Gulf of Mexico. So this is a satellite image. And that's really where most of our funding has been focused, but it's also relevant to other studies of environmental disturbance, other oil spills. So the goal is really to understand how these ecosystems function, understand how things interact in marine sediments. And one thing which we haven't really been asking yet because of the type of data we've been generating is asking whether taxonomic diversity corresponds with functional diversity. And that's true for eukaryotes and also for bacteria. So we want to know that across the breadth of taxonomy. We can also ask whether functional pathways, genes or metabolites, and host microbe interactions are conserved in similar habitats. So does the habitat drive that? And can we identify biomarkers that could be species, it could be genes, or it could be metabolic pathways that allow us to track disturbance and environmental change? And this is particularly relevant from an environmental monitoring standpoint. So whether or not we can give places like the EPA and NOAA tools where they can use genomic information to track disturbance and things like oil spills. And so, as I said, the majority of this work is focused on the Deepwater Horizon oil spill, and I'll talk a little bit about work which we published in 2012 in PLOS One. And so we were really lucky to have collaborators who were in the Gulf region, and they were able to collect pre-spill samples for us. So essentially when the oil started flowing, my collaborator Ken Halanich went down to Dauphin Island, which is where their marine station is. It's a barrier island on the coast of Alabama, and collected samples when the oil started flowing, so before it hit the beaches. And I went back and sampled these sites in September, but the take-home message of this slide is that this oil spill was absolutely massive. This is a cumulative oiling over time, so the well was here, but the oil got absolutely everywhere. I mean, into the marshes of Louisiana, all the way up to the Gulf Coast on the beaches. So it was really a dramatic event that we're still feeling the repercussions of, particularly in the deep sea. Some of the work that's coming out is kind of depressing. All right. So the sites that we focused on for this first study were, again, these 5 sites around Dauphin Island where we had pre-spill samples collected in May 2010, and then I went down and collected post-spill samples in September of 2010 after a summer of a very heavy oiling. And this was using 454 data in conjunction with morphological taxonomy. So we had a nematode taxonomist that was looking at the individual worms alongside us doing the genomic sequencing. And this is, surprisingly, this is actually an impacted beach. So this is a post-spill beach in September of 2010 on Dauphin Island. And this is where I think the power of genomic techniques comes in, because if you went to this beach and you had no idea what was happening in the news, you'd been in a cabin in the woods, and you looked at the beach, you would really have no idea what had happened over the past 6 months in terms of the oil spill. But that appearances are deceiving, and when we actually sequenced the beach sediment, we found a very different story. So despite everything looking fine, we saw this very low diversity community, which ended up dominating this exact beach, or the beach in the last slide. So the pre-spill samples were displaying a typical myofauna assemblage dominated by nematodes, very diverse, lots of metazoa. So that was in May before the oil hit. And then post-spill beaches were showing almost exclusive dominance of fungal taxa in September 2010, despite the beaches looking pretty clean. And so this wasn't just one site. This was consistent across sampling sites, as illustrated by these black bars. So it was a consistent dominance of fungi all around Dauphin Island, and the groups of fungi which were really dominating these communities were Cladosporium and Alternaria. When we looked into the taxonomic literature, there was a little bit of a suggestion that these were potentially either could degrade hydrocarbons or they represented these opportunistic taxa, which could just kind of survive in the most dire of circumstances. So this was our first project focused on the oil spill. And unfortunately, we've been applying for funding for 5 years to continue this work. We just had a little pot of money through NSF to do this work. So we had a lot of questions. It's a very small snapshot of the community. But I mean, the impacts were pretty dramatic. So we were seeing pre-spill sites being very distinct from each other, having a very separate community assemblages, and then this convergence towards a post-spill community where samples were converging onto a very similar set of taxa. And you know, the shifts were just pretty consistent across sites. And this is, again, a unifrac analysis coming out of the CHIME pipeline.
How do you know these aren't seasonal?
Yeah, so we don't. We have collaborators down in Auburn University who have been sampling over time, and what they're finding is that they are getting some recovery of taxa as they continue sampling, but it's a different community set. So the main question that we're trying to ask, or people are trying to ask, is whether or not the recovery of taxa is actually a seasonal thing, so or whether that is a, you know, just a stochastic thing where everything got wiped away, and then the community that recovered is sort of a stochastic accumulation. So there's no baseline information in the Gulf of Mexico, unfortunately, so it's hard to relate that to previous seasonality. But yeah, it's a question that we definitely want to ask. In terms of nematode morphological taxonomy, the patterns were consistent, so the genomic information was validated with the actual the worms themselves, and we found this strong divide again between pre-spill and post-spill sites. So pre-spill sites were dominated by a very high richness of nematode taxa, evenness across genre, and in contrast, post-spill sites showed a different community. So they were dominated mainly by predators and scavenger nematode taxa, and then as well an abundance of juveniles, which potentially suggested some recovery. But unfortunately, we haven't been able to continue the work until now. So as of November 2015, we're lucky enough, I'm co-PI on this grant in conjunction with the University of New Hampshire, Corpus Christi, and the Smithsonian Institution. So this is a new grant from the Gulf of Mexico Research Initiative, where we're going to be continuing this first set of work. So really, we've been waiting around. We have the samples in hand. We just haven't had the continued funding to really delve into this with more sequencing. But now we do. So it's exciting times because this grant is just coming online now. We're getting the paperwork sorted. But we're going to look at genomic responses to the Deepwater Horizon event and attempt to develop high throughput biological assays for oil spills. So we had a lot of samples in hand. A lot of the NSF funding went to me actually going down to the Gulf of Mexico and driving a lot in a rental car all along the Gulf Coast and collecting samples across the Gulf. So we have a mix of pre-spill and post-spill sites. We've also collected time series transects and baseline data from natural oil seeps in the deep sea of the Gulf of Mexico, so we have an idea of what the eukaryotic communities look like at a site where there's oil naturally occurring. And the good thing about the oil spill is that there's a lot of research being done in that area. So there's a huge community built up around the Gulf of Mexico and driven by the Gulf of Mexico Research Initiative, this funding body. So there's a lot of people we can actually ask for samples. So it's pretty easy to go into the community and say, do you have samples from this time in this place? And we're in the process of trying to solicit additional samples to really build up our sample set. But we have hundreds of samples in hand already, so hopefully we'll be able to supplement that with even more samples going forward. And the most exciting thing to me about this grant is the fact that the workflow that we designed. So we're going to be doing this integrated omics workflow where we're finally going to be able to have the money to do targeted genome sequencing of individual specimens. And this ties into the previous research aims, which I outlined in the previous two sections of my talk. So this genome data will be extremely invaluable, not only for this project, but for other research questions and for the community in general. So the goal is to really put it online as fast as possible and make it accessible. And we'll be getting genome sequences by leveraging taxonomic expertise. So we wrote into the grant these workshops where we're going to be having a taxonomist who works on Group A to pick out their specimens, identify them, and then give them to us so we can sequence the genomes. And we'll be doing this in conjunction with environmental surveys where we're just doing bulk DNA sequencing. And then everything's going to inform each other, so we'll be pulling some things through to do shotgun metagenomics to look a little bit at function. And the most important thing is linking known taxonomic IDs with environmental contigs. And it doesn't matter if we have a function for the contig, if we can have a hypothetical protein, but at least if we know it's a hypothetical protein that comes from nematode species A, that's, I think, the most important thing for moving forward, because we can't really do that at the moment. So we'll be leveraging software, which I've worked on, which is the phylocyf pipeline, which is a software tool for doing phylogenetic analysis of genomes and metagenomes. And this is, I worked on the software development team when I was at UC Davis, and this pipeline is great because it allows us to very rapidly mine metagenomic data for specific things that we're looking for. So metagenomic data is very messy, it's hard to deal with, there's a lot of junk in there, but with this pipeline, we can pull out ribosomal RNA reads, we can pull out protein coding genes, so these are single copy nuclear orthologs which have known phylogenetic utility. And my role in this project was to implement the eukaryotic markers for this pipeline. So these are things like elongation factors and tubulin proteins and things which have been used to build phylogenetic trees in protists mainly. And we built, so the phylocyf comes with hidden Markov models implemented already, so you install the pipeline, it has the existing set of HMMs, and you're just putting your data in, and phylocyf does its thing, and goes on to do phylogenetic placement. So that's the crux of this tool, is really putting these marker genes into a phylogenetic topology. And you can do things like get taxonomic summaries, as well as compare samples in a way that you would do similar to CHIME. So you can get a PCA plot, you can look at your taxa in the tree and visualize your data. So in the context of the oil spill, one of the things we can look at is genes that have been found to be relevant for hydrocarbon degradation, such as BSSA, which is a bacterial gene. But another thing I want to do in particular is to develop clade-specific classification markers for nematodes. So part of the rationale for generating genome sequences is to really identify things which will be useful to automatically identify these taxa. Okay. So to summarize, so our initial work focused on the oil spill showed immediate dramatic impacts on sediment microbial communities with a consistent shift across sites. There was a putative disturbance-associated community seen in post-spill sites dominated by fungal OTUs. And unfortunately, eukaryotic research in the Gulf of Mexico has really lagged behind. There's been an expansive sequencing effort for bacteria after the oil spill, and there's a lot of really interesting studies coming out, and full genomes even of these oil-degrading bacteria. But the eukaryotic side of things has not really been pursued by the papers I've seen out there. And we've been lucky enough to have a grant funded which will continue this work and allow us to leverage existing sample sites and ask some of the most pressing questions, really. So as we move forward, we're going to link unknown sequences, ribosomal RNA barcodes, metagenomic contigs to taxonomic IDs for eukaryotes, use hidden Markov models to detect taxon groups and track metabolic pathways, particularly for hydrocarbon degradation, and detect species interactions and associations, hopefully potentially drawing from network biology to look at co-occurrence of taxa, and also find ways to potentially look at symbiotic bacteria or anything else which might be relevant to our understanding of the ecosystem. So this goes beyond just oil spill work. This is also relevant to understanding how sediments function in general. And particularly of interest for the Gulf of Mexico grant is to really pinpoint biomarkers, so either species or genes, which respond strongly to oil spills and environmental disturbance. Nematodes, there is literature out there which kind of says nematodes are very good indicator organisms for things like oil spills because they have a mix of life histories and they have differential tolerances to oil spills, so some species are more sensitive to others. So hopefully we can validate that with molecular data and put forward some sort of standard operating practice for government agencies to use genomic tools. Okay. So that comes to the end of the stories I wanted to tell, and I'll just summarize my research focus for you briefly. So my work focuses on three main areas. Firstly, nematode evolution and phylogenomics, resolving deep phylogeny by focusing specifically on marine taxa with no existing molecular data, Inferring habitat transitions, such as source sink habitats in the deep sea, and looking at the origin of fauna, potential origin. And moving towards comparative genomics and looking to pinpoint evolutionary signatures which enable nematodes to exist in diverse habitats and extreme environments. Secondly, my work also uses high-throughput sequencing to look at biogeography of taxa, asking whether everything is everywhere in marine sediments. Also reassessing classical hypotheses with deep sequencing and trying to get at the big questions in biology. And to do this, we're moving towards visualization tools to novelly explore large data sets. And then finally, the final facet is the systems ecology approach, getting this holistic understanding of what's happening in a handful of mud. And also trying to pinpoint biomarker taxa, which can be used to look at environmental disturbance. So specifically looking at genomic responses to the Deepwater Horizon oil spill, inferring functional pathways and species interactions within sediments, linking reads or contigs to taxonomic IDs, so getting more information out of environmental data rather than just putting it in a database, and advancing computational workflows specifically for eukaryotes, but also broadening that to deal with other types of data and look at other different taxonomic groups, and again, get an understanding of not just a taxonomic silo, but a whole understanding of how these environments function. So with that, I think this research would be well suited to a place like Caltech. I mean, a lot of my work really draws from various communities. So as I said, the human microbiome drives a lot of the methods development. And there's a lot of exciting work in microbial biogeochemistry, I think, which we haven't really capitalized on to study microbial eukaryotes. So in terms of my research here, I mean, I would be extremely excited to leverage the facilities and the expertise here to really make leaps, I think, in some of these research areas. So not just little steps forward, but interdisciplinary work which can really transcend boundaries and do some really exciting science. So leveraging comparative genomics tools, wet labs, computational protocols, and methods from model organism and parasite research in particular. Also in terms of biogeography, linking with the Center for Data-Driven Discovery, I'm always thinking about how the astronomy community deals with things in particular and how they deal with these massive data sets. So I think there's definitely scope to learn from very disparate disciplines and pull tools and discuss common ways in which we can visualize data. Identifying new genomic markers, so other orthologs or gene families which we can look for in these taxa. And in terms of systems ecology, really pulling very strongly from the prokaryotic community. So again, microbial and biogeochemistry. And I've been really interested to use cutting-edge tools such as nanosims to look at very small-scale nutrient cycling questions and interactions between bacteria and eukaryotes. So there's a lot of projects I have currently funded that I work on, which I didn't have time to mention, and I'm happy to discuss any of these. But I just want to finish my talk there, I suppose, and say thank you. Thank you for the invitation. It's my first time visiting here, so I'm excited, and I will take any questions. Thank you. Yeah, so I think the problem is the complexity of the data, so it's very difficult to assemble if you have 100 specimens, taking into account viruses and bacteria, because what we generally find is that most of the data comes from bacteria. So we can, I mean, you can wash. I haven't actually looked at that I mean, you can do the blob plots and things like that, but I suspect it would be at least 50-50, if not more bacteria, just because you can't, I mean, you have the outside and then the gut.
So for some of these organisms that you find more ubiquitously, how do you know that the sites that you're sampling are true habitats as opposed to maybe
Yeah, that's a good question. I think in shallow water sites, that's definitely a concern. I mean, there's speculation that birds can, transport mud, so maybe that explains some of the low genetic divergence. In deep sea sites, I mean, there's really very little transport happening there because everything's sort of slow. So it depends if you're looking at a beach versus the deep sea. But I mean, nematodes, they eat bacteria, so they're definitely in those environments and not being transported in terms of the deep sea. But in terms of shallow water estuaries, there is potential for transport around. But I mean, because of their life histories, I mean, they're definitely there to a certain extent. So I guess it's hard to test the dispersal question.
And another question, how do you know, is there any way of assessing the viability of the nematodes? Because if you look at DNA, you don't know if you're looking at dead nematodes or live nematodes, so maybe evidence that it really is a natural habitat would come from the fact that you can reproducibly sample live nematode population.
Yeah, for the deep sea, the sampling is an issue. We're using a 400 base pair fragment, and most of the ancient DNA and the environmental DNA, in terms of not within a cell, suggests that DNA degrades really quickly. So if you're using a 400 base pair primer set and you're getting data, that suggests that organism is living there, because otherwise the DNA would degrade really quickly. So you could do transcriptomics as well. In the deep sea, it's hard because it's just a sampling issue. I mean, you'd have to use a very sophisticated in situ sampler to inject RNA later, which does exist, but it's hard to get time on those machines. So. And yeah, we have thought about transcriptomics, and we might potentially do that as part of the Gulf of Mexico integrated omics workflow. So doing the metagenomic sequencing, but also metatranscriptomic, also to annotate to help make the gene finding a little bit better. That's a good question. So the work that's come from Europe suggests that there's, not only are there a lot of species, but there's these cryptic species complexes. So there's this one group of marine nematodes, which they have in culture in Europe, and they can do breeding experiments, and usually they have to sort of do the experimental work to differentiate the species, some of the cryptic species, because they are so closely related. And they're also using temporal dynamics. So what happens if you have two closely related cryptic species, you get one increasing in abundance in the spring, and then in the wintertime it sort of flips out. It flips. They haven't extended that to genomic signatures, so maybe if you got the full genome, you'd be able to differentiate those better. So otherwise, if you're using single genes or a couple genes for these taxa, it's just impossible to tell without some sort of experimental work or knowing the life history and the temporal variations. But I'm optimistic it can be solved with genomes, hopefully. So most work happens in prokaryotic microbes, and we know that they turn over nutrients and they have these very specific metabolisms. No one's really done the work on eukaryotes, so I'm convinced they have very important roles. I mean, from a sheer abundance perspective, the fact that they're present in thousands, hundreds of thousands, or millions per meter squared. I mean, you can't have something that abundant and have it not play a role in nutrient cycling. There are nematodes with symbiotic bacteria, so some don't. There's one that doesn't even have a gut. It just lives off its symbiotic bacteria. Some have ectosymbionts as well. There's a Rabia, which is a genus in seagrasses, so it physically moves the bacteria through the seagrass ecosystems, and then the bacteria use the sulfur-rich environment for their metabolism too. So if they're not directly cycling nutrients, then they are facilitating it most likely through the ecology and the life history.
Let's thank Holly again.
Thank you.